it away.
Perfect. Thank you very much for the introduction, Matthew, and for the invitation as well. I'm very excited to um, be here and sharing my project with you guys. So let me get this right. Can you see my screen now?
Yep, we can see your screen.
Yeah, perfect. Okay, so today I'm going to be talking about macrophages, which are one of my favorite cells of the immune system. And so macrophages are myeloid cells that form a very heterogeneous family of immune cells, and they have different origins throughout the lifespan of an organism. They are present in pretty much all the tissues where they have very specialized tissue-specific functions. However, when there is inflammation, these macrophage populations expand dramatically by means of recruitment and proliferation, and they switch their function to a much more common function across all the tissues. And so this functional switch is possible because these cells are very clever and express a vast repertoire of receptors that allow them to sense and uptake things and do something about this sensing. And so they can uptake dead cell material and pathogens either directly or through receptors that recognize these things coated by antibodies. And then in response to these, they can produce antimicrobial mediators. They degrade whatever they uptake, and then they present antigens to T cells and other lymphoid cells. And perhaps more importantly for the talk that I'm going to give today, they also produce anti-inflammatory mediators that allow for the resolution of inflammation to happen. And in particular, it is known that upon recognition of apoptotic cells, macrophages produce prostaglandins, IL-10, and TGF-beta, and all these molecules are very important for the fate and the activity of mesenchymal stromal cells, such as fibroblasts. And so during the tissue repair process, macrophages are present in all the stages of this complicated and orchestrated pathway that the tissue has to go to restore itself. And so throughout this process, macrophages will be sensing and producing different things. And as you can imagine, this whole transition from the beginning to the end is very complicated, and they will have to integrate many signals and activate many genetic modules that will produce different effector molecules. And so to understand something that is so complicated, the macrophage field has proposed this activation paradigm that says that pro-inflammatory cytokines like interferon gamma and microbial motifs like LPS would activate macrophages to produce pro-inflammatory cytokines that will induce further recruitment of monocytes and neutrophils and so on and so forth. When the inflammation declines and the resolution starts, cytokines like IL-4 and IL-13 will be more abundant, and these cytokines will activate macrophages in a different way, in a way that they will induce a very strong proliferation and also the induction of molecules that are involved in the tissue repair process, such as RELM alpha and YM1. And I will ask you to please remember these molecules because they will be relevant for the second part of my talk. The results that I'm going to show you today were the result of a hypothesis that is by no means mine, but rather the result of the thoughts of this very smart woman called Polly Matzinger. And her hypothesis is kind of summarized here. And I have to acknowledge that I stole these schematics from Caetano's talks because I think that is very reductionist, but very effective and very pretty. So her hypothesis said that upon injury, the tissues would release molecules that will alert danger to the immune system, and in this case, macrophages. And macrophages will be able to sense these danger signals because they express different receptors, and in response to that, they will produce effector molecules that will stop the insult and repair the tissue. And I'm interested in all these circuits. I'm interested to know what are the signals that activate macrophages, what are the effector molecules that they produce in response to injury, and what are the mechanisms that allow these macrophages to stop injury and heal the tissue. And so the first story that I'm going to talk about was done when I was a postdoc in Dietmar's lab. And because I was in his lab, I became interested in amphiregulin. Amphiregulin is a, sorry, I forgot to mention. So we're going to start with the response first. And this is what links to amphiregulin. So I was interested in amphiregulin. Amphiregulin is the lowest affinity ligand of the epidermal growth factor receptor, the EGFR. And at that time, there were a lot of prominent papers coming along in high-profile journals showing that amphiregulin is very important for the process of tissue repair. However, the cellular sources of amphiregulin and the mechanism by which amphiregulin mediates tissue repair were not known. And so to address these questions, I decided to use my favorite model, which is this one here. And I am lucky because Juan already explained this model, but I'm going to go over the model again, just for those that were not here last week with us. So this is a model of a helminth infection. The helminth is called Niprostongylus basiliensis. And this little worm is able to infect mice through the skin. And once it's in the skin, it gains access to the circulation. It travels to the lungs, and it gets stuck in these very thin capillaries of the lung, and it bursts these capillaries, and this is how then it gains access to the alveolar lumen, causing more damage to the epithelium. And eventually it crawls up the throat, gaining access to the gastrointestinal tract, where they sexually mature, mate, and produce lots of eggs that are expelled through the feces. These little eggs produce little larvae that are ready to infect again. And in this model, I'm interested in the lung phase. So basically the worms arrive to the lung at day 1 and 2. They arrive almost all together. They cause massive damage. And by day 3, they leave the lung. And then the lung is able to repair itself almost completely in immunocompetent mice. And so we use this model to ask what is the role of amphiregulin here. So I took wild type and amphiregulin knockout mice, and to assess damage and repair, we use we measure oxygen saturation in the blood, normalized by breathing ratio. So as you can see here, from day 1 to 3, there is a massive drop of oxygenation in the blood, which means that there is damage. And this damage is repaired days after infection and is almost restored to normal levels at day 6. And you can see that there's a delay in the recovery in amphiregulin knockout mice. Another clue that told us that amphiregulin knockout mice had delay repair is that they had increased number of red blood cells in the alveolar fluid. And in both readouts, you can see that the delivery of recombinant amphiregulin restores the phenotype or reverses the phenotype of these knockout mice. And so before committing 100% to this project, I wanted to know if macrophages would be important or non-redundant cellular sources of amphiregulin. And so to address this, I infected wild type mice, and I quantified amphiregulin expression of Nipo-infected mice by flow cytometry. And so as you can see in this graph, it's a little bit complicated, but this pink bar here shows that the biggest induction of amphiregulin in various immune cell types occurs in alveolar macrophages. And alveolar macrophages are also the most numerous amphiregulin positive cells at these early stages after infection with the worm. And so based on these, we generated a mouse that lacks amphiregulin specifically in the myelin compartment, We infected these mice, and basically we were able to recapitulate the effect of global deficiency of amphiregulin in both readouts, the oxygen saturation and red blood cells in the alveolar fluid. And so the next thing that we wanted to know is what would be the cues that would drive the expression of amphiregulin by macrophages. And so to address these, I generated loads of bone marrow derived macrophages and treat them with many things that we thought were logical candidates. So I obviously include the factors that induce classical and alternative activation of macrophages, namely LPS, interferon gamma, IL-4, and IL-13. But I also included danger signals such as IL-25, IL-33, TSLP, and ATP. And as you can see here, ATP was the best signal that induced amphiregulin expression by bone marrow derived macrophages. And so to see whether this would be something that could happen in many other macrophages, I isolated macrophages from the lungs and the peritoneal cavities, so real macrophages, resident real macrophages, treat them with ATP. And you can see here that ATP induce a very strong upregulation of amphiregulin in both cell types. And so what these data tell us is that ATP can do it, but it doesn't tell us whether it does it in a physiological context. And so to address this, we infected mice with wild-type mice with the worm, and we deliver apirase to deplete extracellular ATP. So basically, we were depleting the danger signal. And so by doing this, we saw that indeed apirase treatment significantly reduced the expression of amphiregulin by alveolar macrophages, suggesting that indeed the recognition of ATP is an important pathway to induce amphiregulin in these cells. And so having learned all this, now what we wanted to know was the mechanisms by which amphiregulin was promoting tissue repair. And one of the cues that we got from phenotyping the amphiregulin knockout mouse was this one here. So when we were quantifying these molecules, we saw that amphiregulin deficiency rendered mice less able to induce collagen deposition as measured by collagen 1 and collagen 3, and also alpha smooth muscle actin. And so these markers are usually related to induction or a differentiation of myofibroblast precursors into collagen-producing myofibroblasts. And so we hypothesized that the production of amphiregulin by macrophages would target myofibroblast precursors that express the amphiregulin receptor, the EGFR, and allow for the transition of these cells into collagen-producing myofibroblasts. So before we did anything, we already knew that myofibroblast precursors differentiate into myofibroblasts driven by TGF-beta. So TGF-beta is produced by many cells in a latent form that needs to be activated for it to produce its biological effects. And one critical mechanism by which latent TGF-beta is activated is by this complex of integrins that include one beta chain and an alpha V chain. And so this integrin complex, in turn, needs to be activated by something else in a way that is called inside-out activation. And this means that there is a signal, another signal that is recognized by a receptor, and this receptor induces a PLC gamma-mediated activation of a signaling pathway that opens up this integrin, making it more efficient at converting latent TGF-beta into bioactive TGF-beta. And so by doing a series of in vitro experiments that I'm not going to explain because of time constrictions, we were able to show that this signal is actually amphiregulin. So amphiregulin binds to EGFR, its receptor expressed in myofibroblast precursors, And this induces a PLC gamma-induced activation of the integrin alpha V and beta complex, allowing for the conversion of latent TGF-beta into the bioactive form, allowing for its signaling, and this in turn allows for the transdifferentiation of precursors into collagen-producing myofibroblasts. And so what I'm going to show you today is in vivo data that supported these schematics here. So the first experiment that I'm going to show you is this rescue experiment on which we took amphiregulin knockout mice and tried to rescue their phenotype with TGF-beta. And as you can see here, this was the case. So what this indicated is that amphiregulin is working downstream, upstream of TGF-beta. Similarly, we generated knockout that have EGFR deficiency specifically in myofibroblast precursors. And so these mice show the same phenotype as amphiregulin knockouts in the light pink here and here. And so as expected, we were not able to rescue the phenotype by delivering amphiregulin because the receptor is not there. However, we were able to rescue these mice with TGF-beta delivery, as it was the case for amphiregulin deficient mice, again supporting the idea that TGF-beta is acting downstream the amphiregulin and EGFR pathway. The next thing we did, we use a pharmacological inhibitor of the integrin alpha V in wild-type mice. And so we saw that by delivering this inhibitor, we were able to recapitulate the phenotype of amphiregulin EGFR deficient mice. And so TGF-beta was able to revert the effects of this inhibitor, but amphiregulin was not. So again, further demonstrating that amphiregulin is upstream and the final effector molecule is TGF-beta in this pathway. And so this is just to show you that genetic deletion of the integrin alpha V is also phenocopying amphiregulin deficiency and EGFR deficiency. So altogether, what we showed is that upon injury, there is release of ATP as a danger signal. Macrophages are able to recognize this extracellular ATP and produce amphiregulin. And amphiregulin will be then recognized in myofibroblast precursors by means of the EGFR. And this EGFR will induce a signaling that will activate PLC gamma. This will activate the integrin alpha B, beta complex to activate TGF-beta and allow for the transition from precursor to collagen-producing fibroblast. And so now I'm going to switch gears and I'm going to tell you another story that was the result of my PhD. And at this time, I was more interested in macrophages activities during the resolution phase of the tissue repair. And there is many seminar studies showing that during this phase, macrophages need to be activated by IL-4 in order to provide their tissue repair functions. But I wanted to know if they were recognizing something else. And so I was particularly interested in SP-A. SP-A is a large molecule that is secreted or is produced by alveolar epithelial cells and is secreted to the extracellular fluid and is almost exclusively present in there and nowhere else in the body. And so SP-A is able to recognize all sorts of things, bacteria, viruses, parasites, apoptotic cells, and this enhances the uptake of these dangerous materials by macrophages, but it also modulates the activity of macrophages towards pro-inflammatory cytokines and LPS, usually inhibiting their effects. So knowing this, I wanted to know what SP-A would do in IL-4 activated macrophages. And so to do this, I isolated alveolar macrophages and treat them with IL-4 in the presence and absence of SP-A. And this was one of my first experiments of this project, and it was very exciting because you can see that the results were clear. So SP-A was able to enhance IL-4 mediated proliferation of macrophages, and this was measured by incorporation of BRDU. And it was also able to enhance the alternative activation of macrophages as measured by secretion of YM1. And so when we started the collaboration with Judy, she was very excited about this, but she wanted to know more about these potential pathway being activated elsewhere. But as I told you before, SP-A is mainly expressed in the alveolar fluid, so basically only in contact with alveolar macrophages. But SP-A belongs to a big family of soluble collagen defense proteins. And they're called like this because they have a very long collagenous tail that holds their globular heads. In other ways, and in simpler words, it's a family of broccoli-shaped molecules that stick and bind to many, many things. And so it was easy for us to get hold on C1q. And so we repeated this experiment comparing the effects of SP-A and C1q side by side in different macrophages, specifically in alveolar macrophages and peritoneal macrophages. And so what you can see here is that, as I showed you before, SP-A enhanced IL-4-induced proliferation and alternative activation, but C1q had no effect whatsoever. And very excitingly, the opposite was true in the peritoneal macrophages. So SP-A did nothing to the IL-4 treatment. However, C1q enhanced IL-4 effects in terms of proliferation and alternative activation. And so to show that this was not an artifact of an in vitro experiment, we got hold of SP-A and C1q deficient mice and treated these mice with IL-4 in vivo. And so this panel on the left hand is the SP-A knockout mice. And so as you can see here, SP-A deficiency rendered alveolar macrophages unable to fully induce proliferation and alternative activation in response to IL-4, but the phenotype of peritoneal macrophages was fine. Here in the right, I show you the C1q knockout data. And in this case, there was no phenotype for the alveolar macrophages in response to IL-4. However, C1q deficiency rendered peritoneal macrophages unable to fully induce proliferation and alternative activation in response to IL-4. So as opposed to SP-A that is mainly and almost exclusively expressed in the alveolar fluid, C1q can be found in many tissues. And so to know whether C1q would do similar things in other tissues, I analyzed a cohort of mice treated with nothing or with IL-4. And I measured C1q in different tissues. And as you can see here, it was possible for us to find C1q in different tissues, but you can see that C1q was strongly induced in the liver in response to IL-4. So we took our C1q knockout mice again, treat them with IL-4, and check whether we would be able to recapitulate the peritoneal phenotype in the liver. And indeed, this was the case. So C1q knockout mice failed to induce proliferation of liver macrophages and alternative activation of these cells in response to IL-4. So this was all very exciting, but we thought that we were using fairly artificial models in vivo. So we wanted to go one step ahead and try to see whether this SP-A and C1q enhancement of IL-4 effects in macrophages could play a role in a more physiological setting. So I went back to my favorite worm model, infected mice with infected SP-A knockout mice with Nipostrongylus, and then I analyzed the macrophages at day 6, which is when macrophages are alternatively activated by IL-4. And you can see that again, SP-A deficiency prevented macrophages to be fully activated in response to the infection, as measured by realm alpha expression and BRDU incorporation. And this correlated with an enhanced pathology of the lungs in SP-A knockout mice. And it was the failure to heal the tissues in SP-A knockout mice was also observed by the fact that revealed by the fact that they fail to induce collagen production. To do something similar in the liver, we took C1q mice and we infected them with listeria. And this is a gram-positive bacteria that is known to cause extends liver damage. And it is known that the liver recovers itself after infection via IL-4 activation of macrophages. And so again, we saw that C1q knockout mice had deficiency in the activation of, in the alternative activation of macrophages and in the proliferation of Kupffer cells, and these correlated with increased expression of transaminases in the blood, which indicate increased liver damage. And so then we wanted to dig in a little bit into the mechanism by which SP-A and C1q were doing these things to macrophages. And so we started with SP-A. And so we looked at the receptors that have been published or reported to mediate effects of SP-A, and we decided to shortlist these ones that I'm highlighting here, mainly because they are not involved in recognizing bacteria, but they're involved in recognizing cell death. And we thought that this would be more relevant in our setting. And so what we saw is that the blockade of myosin 18A, an unconventional myosin, but not the other two, reverted the effects of SP-A and C1q. So if we start here with the red graphs, we can see that SP-A enhanced IL-4 effects in proliferation and alternative activation in alveolar macrophages that were treated with rabbit IgG, but myosin 18A blocking antibody prevented the effect of SP-A. These are peritoneal macrophages treated with IL-4 plus minus C1q in the presence and absence, in the presence of IgG, rabbit IgG, or the blocking antibody. And again, you can see that the blocking antibody reverted the effects of C1q. And a similar story for liver macrophages treated with C1q. Furthermore, we saw that IL-4 was not only increasing the amount of C1q in the liver, but it was also increasing the amount of C1q in the peritoneal cavity, and IL-4 was increasing the amounts of SP-A in the lungs as well. And it was also increasing the expression of myosin 18A in the cell surface of resident macrophages in these three tissues. So here is the lungs, an increase of myosin 18A, the peritoneal macrophages, and these are Kupffer cells. And so at this point, you might be a little bit confused as to how is it possible that that molecules that have tissue-specific functions are mediating their effects through the same receptor. And so how we interpreted this is that myosin 18A, rather than being a receptor, would be a co-receptor that would associate with different receptors in different tissue resident macrophages that would impart the tissue specificity against SP-A and C1q. Now, another possibility, another explanation for this is that myosin is not a receptor or a co-receptor, but rather the ligand. And so what I think is a possibility is that upon tissue damage, the membrane of many cells will be affected, and this will allow the myosin 18A to be exposed and then recognized by SP-A and C1q. And then this complex of molecules will be recognized by different receptors in different tissue resident macrophages. And this explanation is more obvious in light of another paper that was published back-to-back with ours, showing that the tissue repair properties of IL-4 activated macrophages require concomitant recognition of IL-4, but also of apoptotic cells, cells that are dying. And so in summary, the second part of my talk showed that IL-4 induces this positive feedback loop on which it increases the expression of SP-A and C1q, and also increases the expression of a putative receptor. And the signaling of these receptors synergizes with IL-4 to induce tissue repair molecules that will then recover the tissue after injury. So the take-home message for both parts of my talk is that injury induces the release of many, many molecules that will indicate the immune system that there is some damage going on. In this case, macrophages will have a repertoire of receptors that are transmembrane or soluble that will enable these macrophages to recognize this process of happening, and they will respond by producing effector molecules that will target myofibroblast precursors to induce to differentiate into collagen-producing fibroblasts. And with this last slide, I just want to thank all the people that have taught me so much throughout my scientific career. So first, Christina and Belen during my PhD, Judy, because she was a very important collaborator during my PhD, and her with Lucy Jackson-Jones. They taught me a lot about in vivo experiments. Dietmar, my supervisor of my first postdoc, Caetano, my current supervisor, all the collaborators around the world, the funding, Matthew for the invitation, and you guys for the attention. And so I can take any questions.
Thanks so much, Carlos. That was really cool, really, really interesting. I know you have to head off to your lab meeting, but do you have time for one quick question?
Yes. Let's go for questions.
Okay, cool. So wait, why isn't this? For some reason I can't open the Q&A box. I think, could you stop sharing your screen one moment? I think that might be.
Yes. Can you see it now? I can see them.
Oh, okay. I'll let you read out the question then, because for some reason I can't open it.
Okay. So Mauro is saying, thanks, Carlos, for the great talk. First, some of the papers you cited at the beginning show that IL-6s are one of the main sources of amphiregulin in the lungs and induce tissue repair. In your settings, most of the amphiregulin is produced by macrophages, not much IL-6 contribution. Do you have any thought about that? Does it depend on the infection model? Yes, Mauro, this is a very interesting question. I think that it's not necessarily to do with the infection model, but rather the timing. I think that at the beginning, macrophages would be very relevant sources of macrophages, and later on the course of infection, they could be replaced by other cells, but by this time, these other cells will be performing other actions. The other explanation is the positioning. I think that one of the most prominent injuries using this model is the vascular injury. So the worm basically bursts all these capillaries. So a lot of the damage is vascular damage. And I think that alveolar macrophages are perfectly placed to solve this challenge. But obviously this doesn't mean that other cells can produce amphiregulin and they will go and perform other non-redundant functions. Shall I answer another question, Matthew, or how on time?
Yeah, we can take just one more.
One more, and then you guys can send me the questions through Twitter or mail, and I will answer them all.
And would you put your details in the chat box?
Yes, I will.
That'd be great.
Yeah, I'll put my mail and my Twitter, and then you can send me the questions. Okay, so Eugenio is saying, great talk, Carlos, thank you very much. I was wondering if the ATP is detected by the P2X7 receptor and induces aerobic expression, or is degraded by CD73, CD39 to adenosine, and adenosine is in fact the inducer of aerobic in macrophages. Yeah, this is very, very interesting, and the answer is I don't know. We didn't dig into this any further. So I have more questions, but I think I'll just let Breanna carry on, and I'll answer them through Twitter or email.
Well, thank you so much, Carlos. It was
thank you.
Really cool. Really enjoyed that. So yeah, I'm going to hand over to Sammy a second.
Bye-bye, guys.
See you soon, Carlos.
Bye. Bye.
Hiya. So next up we have Breanna Caruso. I hope I have pronounced that properly. I am terrible with names. She is currently doing her PhD at Oregon Health and Science University in Portland, USA, and her research interests are aimed at better understanding anti-tumor immunity, and ultimately she wants to go on to lead her own research lab. As before, if you want to leave questions during her talk, you can do so in the Q&A box or in the chat box, and we'll ask them all together at the end. Okay.
Thank you so much, Samantha, for that very kind introduction. I'll share my screen here. All right. Okay. Well, thank you everyone for giving me this opportunity to present my work. As Sammy mentioned, I am a rising fourth-year graduate student in the lab of Amy Moran at Oregon Health and Science University in Portland, Oregon. And today I'm going to share with you a sort of side project for my thesis work that is working to better understand the role of PD-1 in T-cell development. And so no talk about thymic development could ever begin without an explanation of thymopoiesis. And so briefly, hematopoietic progenitor cells will enter the thymus at the corticomedullary junction. They undergo a series of developmental stages where they fail to express either the CD4 or CD8 co-receptors and are termed double negative thymocytes. These double negative thymocytes will mature and rearrange their T-cell receptor. And at that point, they will upregulate both the CD4 and CD8 co-receptors to generate double positive thymocytes. Double positive thymocytes are then queried for the formation of a functional T-cell receptor that can recognize antigens presented on self-MHC. And to do this, they will interact with cortical thymic epithelial cells expressing antigens on self-MHC. Thymocytes that have formed a productive TCR will receive survival signals necessary to continue maturation. Otherwise, the cells will die by neglect. It's also at this point that a small subset of cells will develop into alternative lineages, such as NKT cells. And interestingly, NKT cells are actually selected off of lipid antigens presented by other double positive thymocytes rather than interacting with cortical thymic epithelial cells. And so I'm going to come back to this point, so just keep this in your mind that NKT cells are selected in a little bit of a different way in the cortex. Cells that are going to continue on under conventional selection will have upregulated the appropriate co-receptor, either CD4 or CD8 during positive selection, to generate single positive thymocytes that will migrate into the medulla. It is in the medulla that cells are queried for reactivity to self. They interact with medullary thymic epithelial cells that are expressing tissue-restricted antigens, and cells that have generated a T-cell receptor with a low or medium affinity to self will undergo conventional selection to generate things like our CD4s and CD8 alpha beta T cells, and then they will migrate out into the periphery. If, however, the thymocyte has generated a T-cell receptor that has a high affinity for self, it will either be negatively selected for and apoptose, or be diverted down agonistically selected lineages that produce T cells that are important for regulating immune homeostasis, so things like regulatory T cells, and then these cells will then migrate out into the periphery. What I hope that you can appreciate is that signaling through the T-cell receptor is incredibly important for selection. The vast majority of developing thymocytes will fail to produce a productive T-cell receptor and will not receive enough signaling through their TCR to drive positive selection. And then a small subset that have generated TCRs that are reactive to self are going to undergo negative selection. So really there is a small proportion of cells that have been positively selected for, but do not undergo negative selection that are going to go on to develop. And there's a small portion at the sort of upper threshold of T cell signaling that are going to undergo agonist selection. And so since we know that signaling through the T cell receptor is incredibly important for determining which thymocytes are going to go on to develop, what exactly is important for developing the signal strength through the TCR? Previous data has already indicated that signaling through the T cell receptor alone is insufficient to drive positive selection of thymocytes. There are secondary signals that are necessary. This includes co-stimulatory signals through things like CD28. Things like CD28 can interact and combine with signaling through the TCR to drive the activation of various kinase pathways that will allow for the translocation and activation of various transcription factors driving T cell maturation programs. And so we know that things like co-stimulatory molecules are important for driving T cell selection. Is there also a role for co-inhibitory molecules like PD-1? What we know from the periphery is that PD-1, when it interacts with the ligands PDL1 or PDL2, it will inhibit signaling through the co-stimulation and the T cell receptor to dampen activation of downstream kinase pathways and decrease T cell activation. And so when I began my thesis project, I was studying the role of PD-1 in cancer, and we had some interesting questions about what it could be doing in the thymus. Previous reports have indicated that PDL1 and PDL2 are expressed by thymic epithelial cells, with PDL1 being expressed in both the cortex and the medulla, while PDL2 is restricted to the medulla. So one of the first things that I did in this project was identify medullary thymic epithelial cells and cortical thymic epithelial cells in order to query them for expression of PDL1 and PDL2. What I hope you can appreciate is that both medullary and cortical thymic epithelial cells, a small subset, are expressing PDL1, while PDL2 expression is really restricted to medullary thymic epithelial cells, confirming what had been previously reported. And that's quantified here. Interestingly, when I looked at co-expression of PDL1 and PDL2, I noticed that in medullary thymic epithelial cells, most of the cells expressing PDL2 also co-express PDL1. The significance of this remains to be determined. So now we know that the ligands PDL1 and PDL2 are indeed expressed in the thymus, but do the developing thymocytes express the receptor PD-1? I was able to identify thymocyte populations along the developmental pathway and look at PD-1 expression. A small subset of DN4s begin to upregulate PD-1, whereas more mature cells like CD4s and CD8 alpha betas express very low levels of PD-1. Interestingly, some of the highest expressors of PD-1 in the thymus were some of the agonistically selected cell populations, with some of the very highest expressors being regulatory T cell progenitor cells and regulatory T cells themselves, as you can see quantified here. And so when we put all of this data together, we see that PD-1 is actually poised to be able to regulate things like positive selection and agonist selection, whereby double negative 4s have upregulated PD-1 that could interact with PDL1 being expressed by cortical thymic epithelial cells, or interact with PDL1 and PDL2 that are expressed on medullary thymic epithelial cells. And some of the highest expressors are cells that are going to undergo agonist selection. And this led me to wonder if PD-1 was regulating agonist selection specifically. So one of the main questions I have is, what is the role of PD-1 in agonist selection? So I've already talked to you about how there is, it's very important, the signal strength through the T cell receptor, with the very high end of positive selection being where agonistically selected cells will fall. So if we have PD-1 inhibiting both co-stim and the TCR and decreasing the overall TCR signal strength, could we be pulling some of the cells that might have otherwise undergone negative selection into the agonistically selected pool? And so with that, my hypothesis is that PD-1 signaling reduces TCR signal strength to regulate the thresholds that are determining agonist selection. To begin examining this question, I looked at both 3-week-old and 8-week-old mice that were either wild type, deficient for PDL1, or PD-1. I harvested both the thymus and the spleen, dissociated to single cells, and have been analyzing them by flow cytometry, with the hypothesis that disruption of PD-1 signaling will result in an increase in agonistically selected cells. To make sure that thymic development is normal in these animals, I first looked at some of the very earliest developmental stages. I was able to examine some of the earliest stages by identifying DN1s, DN2s, DN3s, and DN4s. What I hope you can appreciate is in 3-week-old animals that are deficient in either PDL1 or PD-1, there really is no difference in any of these cell populations. This is also true at 8 weeks. And this makes a lot of sense if you think about when PD-1 is first expressed. We see PD-1 coming up at the DN4 stage. And so I didn't expect to see any differences in early T cell development since PD-1 is not expressed. When we begin to look at more mature thymocyte subsets, the story is a little bit different. In 3-week-old animals, it seems that there are not big differences in either the CD4 or CD8 alpha beta conventionally selected T cells. However, at 8 weeks, we begin to see an increase, at least in PD-1 deficient animals, in both conventionally selected CD4s and CD8 alpha betas. But my interest is primarily in agonist selection, so I looked at several different agonistically selected cell subtypes. And at 3 weeks, you can see that there begins to trend an increase in the progenitors for regulatory T cells, with a significant increase in regulatory T cells themselves in both animals that are lacking either PD-L1 or PD-1. And at 8 weeks, these differences become somewhat more pronounced. Now we see a difference in regulatory T cell progenitors as well as Tregs themselves. Interestingly, there are no differences in NKT cell selection, either at 3 weeks or 8 weeks old. When I looked in the periphery at splenocytes, I noticed that even though there was an increase in regulatory T cells in the thymus, there are no increases in the periphery. And this could be because there is already a saturated niche in the periphery, and so increases would not be able to be identified. The next thing I wondered is whether or not the effect that I was seeing in the thymus was somewhat dose dependent. And so to do this, I looked at heterozygous littermates of both PD-L1 knockouts and PD-1 knockouts. And what you can see is in the heterozygous littermates of PD-L1 knockouts, they already express the phenotype where there is an increase in regulatory T cell progenitors, as well as regulatory T cells themselves. Interestingly, when I looked at PD-1 animals, I could see that there is a dose-dependent effect of PD-1 knockout, where the heterozygous animals have an intermediate phenotype between wild type and PD-1 knockouts. This led me to the question about how PD-1 actually regulates T cell selection specifically. And so in a paper that came out in Ronald Vail group and Ira Melman's group, they look at PD-1 ability to inhibit different factors. They used a cell-free system where they then expressed the signaling components of various molecules as well as intracellular molecules and expressed different concentrations of PD-1 or CD45. And what I hope you can appreciate here is that CD28 is uniquely sensitive to inhibition and dephosphorylation by PD-1 expression. Here in red, you can see that CD28 is represented on each of these graphs, and it is uniquely sensitive to dephosphorylation by PD-1. This is somewhat counter to what has been thought previously, in that PD-1 was thought to previously be preferentially inhibiting the T cell receptor, but now there is evidence that PD-1 preferentially inhibits CD28. But why is this important? When I look in CD28 knockout animals, we can see there is an increase in CD4 cells with a concomitant decrease in regulatory T cells, meaning that Tregs rely specifically on CD28 for their development. Interestingly, I also saw a decrease in NKT cells, and this has been previously reported in the literature. And so when we think about the development of regulatory T cells compared to NKT cells, these cells are developing in completely different compartments, with NKT cells being selected for in the cortex off of other double positive thymocytes presenting lipid antigens, while regulatory T cells are selected for in the medulla off of tissue-restricted antigens presented by medullary thymic epithelial cells. And so CD28 seems to be critical for the development of both of these cell populations. However, it could be playing different roles. And so with respect to CD28's impact on regulatory T cells specifically, there's some data that suggests that CD28 stimulates FOXP3 expression. And so this is a paper out of Al Singer's group from 2005, where they looked at TCR signaling with or without CD28. And you can see that increasing the TCR signal in the absence of CD28 does not lead to FOXP3 expression. With CD28, increasing the TCR does not impact FOXP3 expression in that it allows FOXP3 to be expressed, but it does not increase with increasing TCR signals. And they were able to look at various factors that are expressed on regulatory T cells, and they saw no big changes when they increased TCR signaling. If, however, you have TCR signaling present but increase the amount of CD28, you see that FOXP3 is uniquely upregulated, along with factors that are expressed on regulatory T cells. And so this led me to wonder what was upstream or downstream of CD28 signaling that could be affecting FOXP3 expression. And I hope you can remember from the beginning of my talk that CD28 stimulates various kinase pathways. And so there is a paper that came out in 2008 that looked at the role of PI3 kinase in Treg development. This is an in vitro assay where the authors either had continuous TCR signaling or TCR signaling for a period of time that was then removed. And you can see that with CD28 and TCR signaling being removed, you can induce a small population of FOXP3 positive cells. However, when you inhibit PI3 kinase or the mTOR pathway, which is downstream of PI3 kinase, you see a vast upregulation of the number of FOXP3 positive cells. And so this indicates that PI3 kinase actually inhibits FOXP3. In another paper that came out was interested in looking at the role of NFAT in FOXP3 expression. And to do this, they generated a luciferase reporter with either no NFAT binding sites or one NFAT binding site or all three NFAT binding sites. And you can see that NFAT binding sites were necessary for the upregulation of the luciferase reporter, indicating its importance in FOXP3 expression. And they further identified that it was specifically PKC theta, whereas active PKC theta allowed for luciferase reporter activity, but inactive PKC theta did not. So together, these data indicate that PI3 kinase inhibits FOXP3 expression, while PKC theta actually promotes FOXP3 expression. So this led me to a new hypothesis, that PD-1 inhibits CD28 specifically to regulate FOXP3 expression through the PI3 kinase and PKC theta pathways. Interestingly, there is data suggesting that PD-1 inhibits CD28 at two important LCK binding sites. The LKC binding site that is primarily dephosphorylated is the one that leads to PKC theta activation. And so this inhibition would mean that there would be a decrease in PKC theta activation, which would allow for the PI3 kinase pathway to be more dominant, leading to inhibition of FOXP3. And it's through this mechanism that I believe PD-1 is restricting Treg development in the thymus. So to begin to understand if signaling through the T cell receptor is being impacted by PD-1, I looked at downstream indicators of T cell receptor signaling. One of these is CD5. CD5 is an inhibitory molecule that is upregulated with TCR signaling and is indicative of the TCR signal strength. So higher levels of CD5 indicate a greater TCR signal strength. And what you can see here is in either 3-week-old or 8-week-old animals, either in PD-L1 or PD-1 deficient, we do not see any changes in CD5 expression. And so that really indicates that PD-1 is not altering signaling through the T cell receptor. And so to briefly summarize what I have shown you here today is that PD-1 is expressed by developing thymocytes, and its ligands, PD-L1 and PD-L2, are expressed in thymic epithelial cells, with PD-L1 being expressed both in the cortex and the medulla, whereas PD-L2 expression is restricted to medullary thymic epithelial cells. Regulatory T cells increase in the absence of PD-1 signaling in both PD-L1 and PD-1 knockouts, and I've shown you that this effect is dose-dependent in PD-1 knockouts. CD28 is absolutely critical for regulatory T cell development, as well as NKT cells. And disrupting the PD-1 signaling pathway does not alter TCR signal strength as indicated by CD5 expression. And with that, I'd like to thank all of the members of my lab, especially my mentor, Amy Moran, my dissertation advisory committee, all of the cores that have been absolutely critical in me being able to perform these experiments, as well as my funding sources. And with that, I'd be happy to take any questions.
That was lovely. What an amazing talk. Thank you so much. There's nothing like a bit of cell signaling to make your Monday better.
Indeed.
So, I am very biased though. Those were some lovely Western blots too. So I have a question that I can ask while people are getting theirs in. Considering the PD-1 inhibitors are now becoming such a huge thing for anti-tumor immunity, do you think there would be differential effects of PD-L1 and PD-1 inhibitors on the process?
Yeah, so this is something that I'm actually particularly interested in. It's a project that I've just begun, is looking at the role of PD-1 and PD-L1 blockade in altering the T cell repertoire in cancer patients. And so I am looking specifically at prostate cancer patients because they are on androgen deprivation therapies that actually allows for thymic regeneration. And so these are a really unique patient population where we can see the impacts of either PD-1 or PD-L1 blockade on T cell development and T cell repertoire. So I, of course, think that, yes, absolutely, there is an impact on T cell development and perhaps repertoire in these patients, but these studies are ongoing. Thank you for that question.
No, it's just as a personal interest of mine, because I work on autoimmunity. One of the major side effects of PD-1 and PD-L1 blockade is autoimmunity. And
Indeed.
that's what I did my fellowship on last year.
Oh, how wonderful.
We were looking at colon cancer patients because they have a similar treatment plan. So it's amazing how worlds cross over.
Indeed.
Has anyone else got any questions? You've got 10 seconds to get them in before I call it. It's like an auction. Cool. In the meantime, if you could pop your Twitter handle into the chat so people can go follow you and ask any more questions if they come up with them later.
Great. Thanks, everyone.
Sorry, one question just came in. You almost escaped, Breanna. So someone has signed in. They haven't put their name up, but said, given the recent evidence for PD-1 in myeloid cells, do you have any data or idea if that's the case for myeloid cell development?
This is another interest of mine. So I have collected data on myeloid cells, and that analysis is ongoing. And so I really can't speak to that as of yet, but it's work that will be coming out. So if you follow me, hopefully you'll see that in the future.
Thanks. I'll hand back over to Sammy.
Perfect. Well, thank you so much for coming along. We will have another seminar again next time next week. I am not with it today. Matt, do you have the schedule?
Yeah, so it will be at 4 p.m. again, GMT. And next week we have Noe Rodriguez and we have Marlene Pinto. 4 p.m. next week. So hopefully see you all there.
Perfect. Bye.
Bye, everyone.